I'm Laura Donlin from Hospital for Special Surgery, here to tell you about our report in JBJS entitled Sequencing of Circulating Microbial Cell-Free DNA Can Identify Pathogens in Periprosthetic Joint Infections. I began this study several years ago upon learning that standard of care culture assays miss pathogen identification in up to 10 to 20% of prosthetic joint infections. As a molecular biologist, I thought we could turn to a phenomenon in the body whereby DNA from dying cells gets processed into a specified size down to about 200 base pairs and circulates as what we now call cell-free DNA in the bloodstream for several minutes. When I began this study, it wasn't actually even clear that bacteria and other microorganisms would have their genome processed in this manner. However, we went along anyway, and I'm now happy to report through a collaboration with HSS and Caries Incorporated that indeed microorganisms causing periprosthetic joint infections do get their DNA processed by the same mechanism. It can be found in our bloodstream and used for pathogen identification for these infections. In our pilot study with more than 50 patients, we were able to increase our pathogen detection rate by using cell-free DNA sequencing from blood from 87 to 94%. Furthermore, we were very interested in whether or not this peripheral blood draw-based assay could be used to track clearance of the infection, and indeed, we were able to show this as well. We are very hopeful to bring this test to the clinic quickly to improve pathogen identification and to be used as a means to track whether or not a prosthetic joint infection has been cleared very soon. Thank you.